So today we're going to talk about meiosis and variation. Apologies about the first slide, it's got a bit of a mess. But causes of variation. So why organisms of the same species might appear different, have different phenotypes, for example. So you have genetic variation as a result of mutation, also meiosis, and we need to understand why meiosis creates genetic variation. There's also the environment, which also contributes towards variation as well. So how does genetic variation make a population stronger? If you've got a greater combination of alleles, different varieties of alleles, so you've got a larger gene pool, then you're more able to survive environmental change. For example, a new disease coming along. Really important terms are diploid and haploid. So here you can see the karyotype of a human. So you've got 23 pairs of chromosomes. So you've got 22 autosomes, and you've got one pair of sex chromosomes here. So this individual will be female, you've got two X chromosomes. You can see that each of these is arranged into homologous pairs. So you've got two chromosome 1s, two chromosome 2s, all the way down to two chromosome 21s down there. Just to remind you what a homologous pair of chromosomes are. So a homologous pair of chromosomes is where you've got the same genes on the same gene loci. So that these have identical genes in identical positions on each of the pair of homologous chromosomes. They have different alleles, so the base sequences will be slightly different, but the genes are the same on each pair of chromosomes. The diploid number is often represented as 2n. So diploid is 2n, haploid is n. It's important you do not learn the definition as half the number. It's one copy of each chromosome. And that's really important you learn as a definition. One copy of each chromosome. That's haploid. So it's important to know that the gametes, the sex cells, that's the sperm and the ova in mammals. In plants, that would be the pollen, the male gamete, and the ova, the eggs, the female gamete. So they are haploid. One copy of each chromosome. So meiosis is what produces your gametes, your haploid gametes. Halves the chromosome number. And it's really important as well because all the gametes are genetically different. So that contributing to genetic diversity, making a population more able to survive environmental change with a greater variety of alleles. So a larger gene pool. It's really important, a question can come up, so why is it important that the gametes are haploid? And that's so when the gametes fuse, and that word fuse is important, so when the sperm and the egg fuse, it restores the diploid number. So that prevents the chromosome number doubling each generation. So you're going for n haploid, the gametes fuse, to 2n diploid. So meiosis involves two nucleate divisions. So this would be a cell with one pair of homologous chromosomes, and this would be an interphase before DNA replication, before meiosis has undergone. So you've got one chromosome, one long strand of chromatin of a father's chromosome, one long strand of chromatin from your mother's chromosome, so one pair of homologous chromosomes. The DNA replicates due to semi-conservative replication. At the beginning of meiosis in the first division, the chromosomes coil up, condense, become visible, and you have a structure where you've got a bit like the same as mitosis, where you've got one chromosome made up from two identical chromatids joined by a centromere. At the beginning of the first nuclear division, the homologous chromosomes pair up, so that forms what's known as a bivalent, and their chromatids wrap around each other in places. That will allow a process of crossing over, which we'll come on to later. So I know there's only one pair of chromosomes here, but in humans, so both chromosome 1s would pair up as a bivalent, both chromosome 2s would pair up as a bivalent, both chromosome 3s would pair up, etc., etc. So in division 1, the spindle fibres would attach to the centromeres, and one from each homologous pair of chromosomes. One chromosome from each pair goes to opposite poles. So the daughter cells now have one copy, so these are now haploid. So these cells are now n, haploid because they've got one copy of each chromosome. In the second nuclear division, it works a bit like mitosis. The chromosomes would then align on the equator. Again, the spindle fibres would attach to the centromeres, split, pulling apart the centromeres, and the chromatids would then go to opposite poles. You end up with 4 haploid daughter cells at the end of meiosis. They would be your gametes, and each of those are genetically different. I know at the minute these don't look genetically different, but with more than one pair of homologous chromosomes, and with crossing over, which we'll come on to in a minute, each of these in the end would be genetically different. They would have different combinations of maternal and paternal alleles. So you can see here on this diagram, just to re-emphasise, here we've got 2 pairs of homologous chromosomes. So these, you can tell these are homologous chromosomes because they're similar size, and these are a pair of homologous chromosomes, they're a similar size. So in division 1, they've formed their bivalence, they've paired up with their homologous partner, they've lined up along the equator, and these 2 will get pulled by spindle to this pole, these 2 will get pulled by spindle to that pole. So in the first division, 1 from each pair of homologous chromosomes go to opposite poles, as you can see by the 2 daughter cells produced. In the second division, works a bit like mitosis, the chromosomes line up on the equator, the spindle fibres again would attach to the centromere and pull the chromatids apart. So the chromatids would move to opposite poles. And you end up with genetically different haploid gametes, and 4 of them at the end. You do also need to be able to compare this with mitosis in a compare and contrast question. An example is in the long answer question pack. So the 2 most important things you need to understand about meiosis is, first of all, be able to describe the process, which we've just talked about, and secondly, how meiosis causes genetic variation, and it's 2 answers, crossing over and independent segregation. So we're going to go through how that happens now. So here we can see, so in crossing over, we've got our bivalence form. So in prophase 1, this is a homologous pair of chromosomes, formed a bivalence, so they're paired up. The sections of chromatids cross over each other at a point called a chiasmata. You then have equal lengths of non-sister chromatids exchanging, so the alleles are exchanged. So if this was the maternal chromosome from the mother, containing your mother's alleles, this is the paternal chromosome from your father, containing father's alleles. You can see now on this chromatid, it's not all got alleles from the mother. There's a section that's got alleles from the father, and this chromatid's not all got alleles from your father, it's got a section that's got alleles from your mother. So we end up with chromosomes with new combinations of alleles being produced. And here you can see this happening from a microscopic slide. The second way that genetic variation can be introduced in meiosis is through independent segregation. This is random alignment of chromosomes along the equator. So ultimately produce gametes with different combinations of maternal and paternal chromosomes. So in here I've drawn the equator, and you can see that on here you'd have all maternal alleles being pulled to that pole and all paternal alleles being pulled to that pole. You can see with the 2 gametes that are produced. However, it's completely random which of the homologous chromosomes from each bivalent lines up on each side of the equator. So because these have arranged this way on this one and the opposite way on this one, you end up with different combinations of maternal and paternal chromosomes with their respective alleles in each gamete. So we saw in the first picture where you've got 1 pair of homologous chromosomes. So we've got 1 pair of homologous chromosomes. We had 2 different types of gamete produced. Here we've got 2 pairs of homologous chromosomes, and we've got 4 different combinations of gametes produced. In humans, we've got 23 pairs of chromosomes. Just from independent segregation, how many different combinations of gametes could be produced? So the formula is 2 to the n, with n being the number of pairs of chromosomes. So it'd be 2 to the 23. That's a huge variety. And then this, coupled with crossing over as well, would result in billions of different combinations of gametes that could be produced from meiosis alone. This isn't a process of meiosis that introduces variation, but it is another source of genetic variation. Yes, you produce your gametes, they're all genetically different, but it's completely random, for example, which sperm fertilises the egg. So we're going to look at two very common long answer questions to do with meiosis, and this will assess to see if you've understood the main mark points. So the first one is, how does meiosis produce gametes that are all genetically different? And here's the mark scheme with that. So you've got crossing over, where alleles are swapped at chiasmata between homologous chromosomes, and that produces new chromosomes with new combinations of alleles. Second way is independent segregation, where it's random alignment of chromosomes on the equator, where the bivalents line up. And that produces gametes with different combinations of maternal and paternal chromosomes. Second common question is, describe what happens to chromosomes in meiosis. And this would be the long answer question that would go through this. There's a lot of things that happen on here, and some of these are similar to mitosis, some of them are different, so you would have to be able to do a compare and contrast question as well. So we can see here, initially the chromosomes shorten, thicken, condense, they become visible, just the same at the start of mitosis, where you've got your chromosomes made from identical chromatids joined by a centromere. What's different though is that at the start of meiosis they form bivalents, the homologous chromosomes pair up. Also different to mitosis is at the beginning, in start of prophase, you've got crossing over, so you get chiasmata or chiasma plural forming between the homologous chromosomes. Similar to mitosis, you've got the centromeres attached into spindle, and it is the spindle contracting that's going to move the chromosomes. And again, similar to mitosis, this would be at the equator of the cell. The first division, the homologous chromosomes move to opposite poles, so that is different to mitosis. And in the second division, the chromatids are separated. So that's similar to what happens in mitosis. Spindle splits the chromatids. They're not identical in meiosis though, due to crossing over. Another difference is meiosis has two divisions, whereas mitosis has one. So now we're going to look at two trickier application questions. So we just have a quick think about this first. Now, without crossing over, you can see how gametes, remember, are haploid, so you have one copy of each chromosome. So in each gamete, so at the end of meiosis you can have these different combinations. So gametes are haploid, so you've got one copy of each chromosome. So at the end of meiosis we have one chromosome that's long and one chromosome that's short, chromatid that's short in each cell. And due to independent segregation, similar to the picture where we had two pairs of homologous chromosomes before, due to independent segregation you can end up with four different gametes, variety of gametes produced at the end. This question is a bit different. So if we just have one pair of homologous chromosomes, what would you expect to happen? Well, if you imagine that eventually you're going to split the chromatids apart in the second division. So in the first division, this chromosome went to that pole, in the first division this chromosome went to that pole. So you've got a daughter cell here and a daughter cell here. Normally they would be split, and the chromatids in the second division, so you end up with one daughter cells that either they've only got big G, big D, and other daughter cells have only got little g, little d. like this. Apologies for my scruffy writing, it's hard on the iPad. So how have we got a situation where we've got some daughter cells that have got this combination with a big G little d or a little g big D? And it's to do with crossing over. But the chiasmata must form, so here they would form a bivalent, the chiasmata must form between the G and the D. So this part would break off and join this chromatid, so you end up with a little g big D on this chromosome, and this big G will break off and rejoin here, so you end up with a big G little d on that chromatid, like this here. And then ultimately these would separate, and you could end up with, in the second division of meiosis, and this would be the 4 different gametes that could be produced at the end. So you've got more variation due to crossing over, the chiasmata forming between genes G and D. So why is there less of these than there are those? And it's because crossing over is a rare event.